Hello students, myself Bhanu Solanki, Senior Faculty Sri Chaitanya, Department Botany. Here there in this program we will discuss some of the chapters and their specific areas where there you have to recall them properly so that in exam the things can remain vital for you. So today's chapter is Anatomy of the Flowering Plant. It is a very significant chapter. Here in this chapter, as we know that according to the new syllabus of NTA, we are going to discuss the content. Anatomy of the flowering plant means a study of the internal organization. Different organs in the different plant show differences in their internal structures. It means anatomy of the root, anatomy of the stem, anatomy of the leaf are different. Why they are different? Because plant have to introduce a certain kind of adaptations. So these adaptations means to achieve a particular function, to achieve in different kind of environmental conditions what a plant structure have to carry the set of tissues so that that function can be achieved or performed in a perfect manner. For this purpose, there is the differences in the internal structure or organization of the root, stem and leaf. Different plant organs have different internal organization. Similarly, same plant organ in the different habitat also have a different internal organization. So internal organization is one of the strategy with help of which plant can achieve their best survival conditions. There is a content about the tissue system. We have discussed types of tissues based on the type of cells present, means simple tissue, permanent tissues, complex tissues, like that. If made up of the similar kind of cells, so we call them simple tissues, and if made up of the different type of tissues, so then we will call it different type of cells, then we will call it complex tissues. Xylem and phloem both are complex tissue, while parenchyma, collenchyma, sclerenchyma are the simple permanent tissues. So tissues are very depending on their location in the plant body. Means where they are located, accordingly they are variable. The tissues vary depending on their location into the plant body. Their structure and function also dependent on the location. Means what tissue will perform what function and it would carry what kind of a structure, it is decided by the location. So these are very important crucial fact of the NCERT that you have to keep in your memory. On the basis of their structure and location, there are three types of tissue systems. Based on the structure and location, there are three types of tissue systems: epidermal tissue system, ground tissue system, and ground tissue system, that is also called fundamental tissue system, and vascular or conducting tissue system. The epidermal tissue system, first of all, we are exploring. The epidermal tissue system forms outermost covering of the whole plant body, comprises epidermal cells. Means what epidermal tissue system include? Epidermal cells, stomata and the epidermal appendages. These are the three main crucial things: epidermal cells, stomata and epidermal appendages. What epidermal appendages includes? Trichomes and hairs. Epidermis is the outermost layer of the plant body, made up of the elongated, compactly arranged cells. Cells of the epidermis are compactly arranged, which form a continuous layer. Epidermis is usually a single layered. Exceptions are there, like in hydrophytes or xerophytes, where their epidermis may be more than one layer. Epidermal cells are parenchymatous. You have to keep this also in your memory, that the epidermal tissues are parenchymatous cells. They have a small amount of cytoplasm lining to the cell wall and a large vacuole. The presence of large vacuole is also one of the characteristic feature of these members. The outside of the epidermis is often covered with a thick waxy covering, and that is called cuticle. Remember, cuticle is not present in underground plant parts. Means cuticle we can observe only on stem and leaves. Which is what it is doing? It is preventing to the loss of water. Cuticle is also absent into the roots, as I mentioned earlier. Stomata are present into the epidermis of the leaves, sometimes on the stem also when it is a primary body, but they are also absent in root region. Stomata regulate to the process of transpiration and gaseous exchange. Means both transpiration and gaseous exchange. These are the two things controlled by our stomata. Each stomata is composed of the two bean-shaped structures, yani bean-shaped guard cells if it is a dicot, and if monocot then dumbbell shape. So known as the guard cells which enclose to the stomatal pore. In grasses mean monocot, the underground cells are dumbbell shaped structure. Outer wall of the guard cells, this is very important fact, outer wall of the guard cells is thin and the inner wall of the guard cells is highly thickened. So you should keep it also in memory. The guard cells possess chloroplast and regulate to the opening and closing of stomata. It means chloroplast of the guard cell is associated with the opening and closing of stomata. Sometimes a few epidermal cells into the vicinity of the guard cells are specialized parenchymal cells and they are known as subsidiary cell. Now together, what? Stomatal aperture, yani pore, guard cell and subsidiary cell. They are collectively known as stomatal apparatus. They are collectively known as stomatal apparatus. So stomatal apparatus include three things: stomatal aperture, guard cells and subsidiary cells. Here there, this diagram with labeling you have to keep in your memory. Although here chloroplast shown, here not shown, but it doesn't mean that the chloroplast are not present into the guard cells of the monocots. They are present equally in both guard cells of dicot as well as guard cells of monocot. The outer cells of the epidermis bear a number of hairs. Root hairs are always unicellular. They are elongations of the epidermal cells and what they are doing, helping into the water and minerals from the soil. On the stem, epidermal hairs called trichome. Hairs of the stem are known as trichomes. Trichomes of the shoot system are usually multicellular. So this is the difference. Root hairs are unicellular while stem hairs are multicellular. They may be branched, they may be unbranched, they may be soft, they may be stiff or they may be secretory also. What is the function of trichome? They are preventing the loss of water. Preventing water loss is the main function of trichomes. Then second is a ground tissue system. All tissues except epidermis and vascular tissue, vascular bundles constitute to the ground tissue system or the fundamental tissue system. Very important thing, ground tissue system is consist of, you have to keep it in your memory, ground tissue is consist of simple tissues such as parenchyma, collenchyma and sclerenchyma. Ground tissue is made up of parenchyma, collenchyma and sclerenchyma. Epidermal tissue was made up of only parenchymatous cells, but here ground tissue include all three types of permanent tissues. Parenchymatous cells of the ground tissue we can see into the cortex region, in the pericycle region, in the pith region and in the medullary range. They all are representative of ground tissue and are made up of parenchyma. Condition is that in the primary stem and primary root. Means what is the question? Parenchyma constitute ground tissue in primary root, yes. Primary stem, yes. Secondary root, no. Secondary stem, no. Only in primary root and primary stem, ground tissue ground tissue is made up of the parenchyma. Ground tissue of the leaves, inside the leaf, the ground tissue is consist of the thin-walled chloroplast containing cells and we know that what these cells are known as mesophyll. So ground tissue of the primary root parenchymatous. Ground tissue of the primary stem, parenchymatous. Ground tissue of the leaf is also parenchymatous, but it is called mesophyll. Next is a vascular tissue system. The vascular tissue system consists of the complex tissues. Always it is made up of complex tissues. Consists of complex tissues. What are the complex tissues? Phloem and xylem. The xylem, phloem together constitute to the vascular bundle. In the dicotyledon stem, cambium is present between the phloem and xylem. In dicot stem, cambium is present in between the xylem and phloem, and such type of vascular bundles, because of the presence of cambium, possess the ability to form secondary xylem and phloem tissues, and hence they are known as the open vascular bundle. So open vascular bundle is characteristic of the dicot stem. In the monocotyledons, the vascular bundle have no cambium present, so their vascular bundles are known as closed vascular bundles because they cannot add secondary xylem and secondary phloem. When xylem and phloem within the vascular bundle are arranged in an alternative manner, means xylem somewhere else, phloem in somewhere else, means different radius. So such type of vascular bundles are known as radial vascular bundle. Radial vascular bundle is a characteristic feature of roots. Conjoint type of vascular bundle, the xylem and phloem are joint situated on the same radius. It means you can say open and closed vascular bundles are always conjoint vascular bundle, or you can also call conjoint vascular bundles are open and closed types. Such vascular bundles are common in the stem and leaves. So conjoint vascular bundle means xylem and phloem present on same radius are characteristic of both stem and leaf, while radial vascular bundles are characteristic of the roots. The conjoint vascular bundle usually have phloem located on the outer side of the xylem. Usually, yani majority of cases, in most of the cases, primary phloem would be present towards the outer side and primary xylem would be present towards the inner side in case of conjoint type of vascular bundle. Whenever primary phloem is present towards the outer side and primary xylem is present towards the inner side on the same radius, such type of conjoint vascular bundles are known as collateral vascular bundle. So most of the time conjoint vascular bundles are generally collateral. These diagrams also you have to keep in your memory. Anatomy of the dicotyledonous and monocotyledonous plant means what are the differences between dicot root and monocot root, dicot stem and monocot stem, dicot leaf and monocot leaf. Dicotyledonous root. It shows transverse section of the sunflower root, means we are taking case study of the sunflower. Outermost layer epidermis. Epidermis of the root is known as epiblema. Means term epiblema we can apply only for the epidermis of root, not for the epidermis of stem, not for the epidermis of leaves. Many of the cells of epiblema protrude in form of the unicellular root hairs, as we know. The cortex is consist of the several layers of thin-walled parenchymatous. So here also you have to keep in memory that the cortex region is made up of parenchymatous cells. The innermost layer of the cortex is known as endodermis and it comprises a single layer of the barrel-shaped structure. So here you have to keep in memory cortical cells, the innermost layer of the cortical cell called endodermis. Endodermis is a single layer barrel shape. The cells of the endodermis are barrel shape. And without any kind of intercellular space, the tangential wall as well as radial wall of the endodermis cells have a deposition of the water impermeable waxy substance material, that is suberin. So suberin is a water impermeable, it is a waxy chemical, it remains present in the endodermal cells. On which wall? On the tangential wall and on the radial wall. This deposition of the suberin called Casparian strips, the presence of the Casparian strip is the characteristic feature of endodermis. True endodermis means Casparian strips must be present. Next to the endodermis lies a few layers of the thick-walled parenchymatous cells, that is called pericycle. Initiation of the lateral root and vascular cambium during secondary growth. Two functions are mentioned here. Initiation of the lateral roots and development of the vascular cambium during secondary growth are the two functions of pericycle. In dicot root, pericycle perform two functions. One is the initiation of lateral root and second is the secondary growth. The pith is small and inconspicuous. Pith is small and inconspicuous. The parenchymatous cells which are present between xylem and phloem are known as conjunctive tissue. Means conjunctive tissue is a characteristic only of root. In between the primary xylem and primary phloem, there is a parenchymatous cell, and these parenchymatous cells are constituting to the conjunctive tissue. So keep in memory, conjunctive tissue term you have to use only for root, both dicot root and monocot root. There are usually 2 to 4 xylem and phloem patches are present. When 2 xylem and 2 phloem patches present, it is called diarch. If 3 xylem, 3 phloem, triarch. If 4 xylem, 4 phloem, tetrarch. So in dicot root, there is the diarch to tetrarch condition, means 2 to 4 xylem and phloem patches are present. Later a cambium ring may develop between the xylem and phloem. All tissues on the inner side of the endodermis, from endodermis to inside, means pericycle, conjunctive tissue, primary xylem, primary phloem, pith, they are collectively known as stele. All tissues lying inner to inner endodermis are collectively known as stele. So what is the composition of stele? It includes pericycle, it includes conjunctive tissue, it includes primary xylem, it includes primary phloem, and it also includes pith. Then anatomy of the monocot root. The anatomy of the monocot root is similar to the dicot root in many respects. I just want to highlight what are the differences. Number one, in case of monocot root, pericycle will perform only one function, that is initiation of lateral root. Means cambium ring development would not be there because monocot do not have provision for secondary growth. Second thing, number of xylem and phloem patches are usually more than 6. As mentioned here, there are usually more than 6 polyarch condition we will observe here. So usually more than 6 polyarch xylem bundles are present. In dicot root we have discussed diarch to tetrarch, here hexarch or more than that. Pith is here large and well developed. Where it was there small and inconspicuous, here it is a large and well developed. So, and there is no secondary growth. So we have highlighted 3 major differences in the anatomy of the dicot root and monocot root. Dicot root, pericycle perform 2 functions, monocot root, pericycle perform 1 function. Dicot root, pith is small and inconspicuous, monocot root large and conspicuous. Dicot root, diarch to tetrarch condition, here hexarch or more than 6 xylem and phloem patches we will observe. These are the 3 fundamental differences. So by counting to the number of xylem and phloem patches, you can easily distinguish or judge that the given diagram is a dicot root or monocot root. Rest all other features are same. Then anatomy of the dicot stem. The transverse section of the typical young dicotyledonous stems is shown that the epidermis is outermost protective layer as usual, covered with a thin layer of cuticle, we have discussed already. It may bear trichomes and few stomata also. Few stomata also may be present on stem condition on the primary state. The cells arranged in multiple layers between the epidermis and pericycle constitute to the cortex. It consists of 3 subzones. Outermost hypodermis, which is consists of the layers of collenchyma. Hypodermis of the dicot stem is made up of collenchyma, followed by general layers of the cortex, which provide, this collenchyma is helping with what? It is providing mechanical support. Because collenchyma is a living permanent tissue, so it is It helps into the providing mechanical support. Because of this reason, dicot stem have collenchymatous hypodermis. Cortical cells below hypodermis consist of the rounded, thin-walled parenchymatous cells with conspicuous intercellular space. The innermost layer is of the cortex called endodermis, but endodermis of the dicot stem is different from the roots. How? The cells of endodermis are rich in starch grains and layer, and the layer is also referred as the starch sheath. Means there was the Casparian strips in dicot root and monocot root. Instead, in dicot stem there is the starch sheath, means there is no Casparian strips. This is diagram, label diagram you have to keep in your memory. Now after endodermis, above the phloem in form of the semilunar passage, of the sclerenchyma, what is present? Pericycle is present. So pericycle in dicot stem up just above the phloem is in form of the semilunar passage and it is made up of sclerenchyma. In between vascular bundles there are few layers of radially placed parenchymatous cells are present that we are calling as medullary ridge. A large number of vascular bundles are present and they remain arranged into the ring. Vascular bundles of the dicot stem are present in form of the ring. The ring arrangement of the vascular bundle is characteristic of the dicot stem. Each vascular bundle is conjoint, means xylem phloem present on the same radius. Open, it means cambium present in between the xylem and phloem, and with enlarged protoxylem. When protoxylem facing towards the center and metaxylem facing towards the periphery, this is called enlarged condition. So xylem of the root is exarch while xylem of the stem is enlarged. A large number of rounded parenchymatous cells with the large intercellular space is occupying into the central position that we are calling pith. So pith in dicot stem is also well developed and distinguishable. Then monocot stem. The monocot stem has sclerenchymatous hypodermis. This is one of the difference we have already discussed. Sclerenchymatous hypodermis. While you remember in dicot stem it was the collenchymatous. A large number of scattered vascular bundle. Vascular bundles, unlike dicot stem, in monocot stem they are scattered. Each is surrounded by sclerenchymatous bundle sheath. That character was missing in dicot stem. Vascular bundles were not covered with bundle sheath, but here they are covered with bundle sheath, and that bundle sheath is made up of sclerenchyma. And the large conspicuous and parenchymatous ground tissue is present. Vascular bundles are conjoint as usual and closed, unlike dicot stem where there it was the open. Peripheral vascular bundles, this is very important line. Peripheral vascular bundles are smaller and the centrally located vascular bundles are larger one. So vascular bundle towards the periphery are small size and vascular bundles which are present on the centers, they are relatively larger size. Phloem parenchyma is absent, very important factor. In monocot stem, phloem parenchyma is not present, but water containing cavities are present within the vascular bundles. So in this manner we observe so many differences in between the dicot stem and monocot stem. In dicot stem, hypodermis was collenchymatous. In monocot stem, it is sclerenchymatous. In dicot stem, endodermis was present in form of starch sheath, but in monocot stem, there is no starch sheath or endodermis. In monocot stem, except hypodermis, neither there is an endodermis, nor there is a pericycle, nor there is a medullary ridge, nor there is a pith region. Vascular bundles are uniform size, vascular bundles of different size. Dicot stem, monocot stem. Vascular bundles not remain surrounded by bundle sheath, but here vascular bundle remains surrounded by sclerenchymatous bundle sheath. Vascular bundles remain arranged in a ring while vascular bundles are scattered. Vascular bundles are conjoint collateral open. Vascular bundles are conjoint collateral closed. These are the main fundamental differences. Phloem parenchyma present here, phloem parenchyma absent there. So in this manner, you should keep just only these fact or important points of the differences in your mind. Then there is the anatomy of the leaf. Dorsiventral leaf means dicotyledonous leaf. When inside a leaf, dorsal surface and ventral surface are clearly visible and distinguishable, they are known as dorsiventral leaf. In the vertical section of the dorsiventral leaf, through the lamina show three main parts, namely epidermis, mesophyll and vascular tissue system. Epidermis, mesophyll and vascular tissue system. Epidermis, which cover both upper surface and lower surface. Upper surface, upper means adaxial, adaxial means ventral. Adaxial means ventral, abaxial means dorsal. So upper surface means adaxial, yani ki ventral epidermis, and the lower surface means abaxial, abaxial means dorsal surface of the leaf is conspicuous and remain covered with the cuticle. Abaxial, yani ki dorsal epidermis generally bear more number of stomata. It is an adaptation so that the more number of stomata remain present on lower epidermis, means dorsal surface means abaxial epidermis. So this fact also should keep in memory. More number of stomata, on abaxial surface as compared to adaxial surface. This type of condition where more number of stomata present on lower surface and less on upper surface, this is called hypostomatic condition. Later may even lack stomata, means on the ventral surface sometimes even there is no stomata. The tissue between the upper and lower epidermis is called mesophyll. Mesophyll, which possess chloroplast and carry out photosynthesis. It is made up of the parenchyma. It has two types of cells. What are these two types cells? Spongy parenchyma and palisade parenchyma. Palisade parenchyma and spongy parenchyma. Adaxially placed palisade parenchyma means it is present on the upper epidermis side. The adaxially placed palisade parenchyma is made up of elongated cells which are arranged vertically and parallel to each other. The oval or rounded and loosely arranged spongy parenchyma is situated below the palisade parenchyma and extend up to the lower epidermis. Means just beneath from the palisade to lower epidermis, the entire volume is occupied by spongy parenchyma. There are numerous large spaces and air cavities between these cells. Vascular tissue system include vascular bundles which can be seen in the veins and the midrib. The size of vascular bundles are dependent on the size of veins. This is important point again. The size of vascular bundles dependent on the size of veins. The veins vary in the thickness. The veins vary in the thickness into the reticulate venation of the dicot leaves. Means in dicot leaves, these veins and venlets form a network and that is called reticulate venation. The vascular bundles are surrounded by a layer of thick walled bundle sheath cells. Means bundle sheath here also is a characteristic. So here what we are observing that the mesophyll is differentiated into the palisade parenchyma located towards the adaxial means ventral surface, long elongated compactly arranged cells, and second is spherical or oval shaped loosely arranged spongy parenchyma. Vascular bundles are present. Vascular bundles are here surrounded by bundle sheath. That is the important feature. Size of the vascular bundle is dependent on the size of the veins and midribs. Then next is a isobilateral leaf, means monocotyledonous leaf. The anatomy of the isobilateral leaf is similar to that of the dorsiventral leaf in many ways. It shows following characteristic differences. What? In isobilateral leaf, stomata are present on both the surface equally. This is one of the difference. Stomata are present on both the surface. Second difference what we are watching, that Mesophyll is not differentiated as parasite and spongy parenchyma. These are the two main differences that you will notice in the anatomy of dorsiventral leaf and isobilateral leaf. In grasses, certain adaxial epidermal, adaxial means again ventral. So I highlight it again. In grasses, certain adaxial epidermal cells along the vein modify themselves into the large, empty, colorless cells. These cells are known as bulliform cells. When bulliform cells of the leaves have absorbed water, means they are turgid, and are turgid, the leaf surface is exposed, and when they become flaccid, yani lose their water content due to the water stress, they make the leaves curl inward, yani rolling of the leaf. So when leaf get rolled, so its surface area will decrease. As its surface area decrease, so you can understand that the loss of water will minimize. The parallel venation in monocot leaves is reflected in nearly similar size of the vascular bundles. As we know that in case of monocot leaves or isobilateral leaves, their vascular bundles are of almost uniform size. Why? Because all the veins and lateral veins are of equal size. So you can say that parallel venation is a characteristic of isobilateral leaf, and reticulate venation was the characteristic of dorsiventral leaf. So in isobilateral leaf of the monocot, all vascular bundles are nearly of same size. It reflects to the parallel venation of the leaves. So I repeat one more point here there. In dorsiventral leaf, venation was reticulate, and all the vascular bundles are not of equal size. But in case of monocot leaf or isobilateral leaf, venation is parallel, and this parallel venation can be reflected in terms of the uniform size, nearly uniform size of the vascular bundles. So this is all about this chapter of anatomy. In this chapter, while attempting a question, direct question asked from the NCERT region, as in case of the NEET 26 also there were some questions directly they asked. So you need not to be panic about this chapter. Just go through the NCERT, and especially those points that I highlighted here, you just revise once. I can assure you that from this chapter, every question whatever asked by the RE-NEET exam, you would be 100% in a comfortable zone, and you will do it correctly. Just only keep one thing in mind, that read the question properly, means dicot is written or monocot is written, dicot leaf is written or monocot leaf is written, dicot stem is written, monocot stem is written. Because generally what happened, once we see dicot, we didn't see the another word, that they are asking about the stem, or they are asking about the root, or they are asking about the leaf. So while reading a question, make sure that what is given there, dicot root, dicot stem, dicot leaf, monocot root, monocot stem, monocot leaf, means accordingly you have to take a decision. So while reading a question, don't be in hurry, don't do hasty reading, just read question properly, make sure about what they are asking, accordingly your answer would be there among the given choices. In such type of a paper, attempting a question and reading wrong would definitely be very much harmful and very much injurious. Why? Because no other student will make such kind of a mistake. But it is a common thing that we should take this precaution while attempting a question, that titles given in a question should be properly read. As I said, dicot, monocot, root, stem, leaf, etc. So you have to prepare it accordingly. Thank you very much. Wish you all the very best.